Everyone. Welcome to the 2025 NDARC webinar series. My name is Calvert Tisdale. I am a postdoctoral research fellow at the National Drug and Alcohol Research Centre, and I will be the chair of this webinar today. So this is my first webinar in 2025, and I would like to start by welcoming our audience. That includes people from many diverse and valued perspectives and experiences, including members of community, health professionals, researchers, other stakeholders, and everything in between and otherwise. Before we start with the webinar, I would like to firstly begin by acknowledging the Bidjigal and Gadigal people as the traditional custodians of the land from which I'm joining this meeting today. I'd also like to acknowledge the traditional owners of country throughout Australia, noting that our speaker, panelists and audience are joining from many different locations, not only across Australia, but internationally as well. And I'd also like to pay my respects to the elders past and present and emerging, and extend that respect to any First Nations people present today. So today I have the distinct pleasure of introducing Professor Valentina Lorenzetti, who is the lead of the Neuroscience of Addiction and Mental Health Program and founding Deputy Director of the Healthy Brain and Mind Research Centre at ACU. Her research aims to identify the core mechanisms of addiction and target them using novel interventions through advanced multimodal MRI, global multi-site studies, and international consensus on new metrics. Today, we are lucky to have Professor Lorenzetti present an overview of the research surrounding the neurobiology, risks, and potential of cannabis use. And we will also luckily be joined for the panel discussion by Emeritus Professor Wayne Hall. Wayne is an Emeritus Professor at the National Centre for Youth Substance Use Research at the University of Queensland and is a leading expert on substance use and cannabis use specifically, having advised the WHO or World Health Organization on the health effects of cannabis use, the effectiveness of drug substitution treatment, and the contribution of illicit drug use to the global burden of disease. So we are very lucky to have him join us later today. We're going to have time for questions at the end, so please send them through the Q&A function, not the chat function, and I will present as many as possible given the time to Wayne and Valentina at the end of the presentation. I'll also do another reminder, but please put your questions in that Q&A tab. So now I would like to invite Valentina to please turn on their camera and take it away. Thank you so much, Valentina.
Well, thanks so much for having me here, Calvert and Dark. It's an absolute honor to be here, and I'm really excited to share some of our research. And thank you, Wayne, so much as well for being here today and for supporting the work. I'm looking forward to the discussion at the end of the presentation. So the first thing I wanted to start with is I have no conflict of interest to declare. And today the talk is going to be split in two parts. The first part is about metrics of cannabis use, which seemed to be a very boring topic, but actually it became something quite useful and quite discussed. So I'm looking forward to share some of the findings with you. And the second part covers a little bit on the evidence that our lab has accumulated over time on the neurocircuitry of cannabis use disorder, and some new interventions that we've been trialing to see if we can mitigate some of the neural alterations we have been reporting in people with a cannabis use disorder. So thanks, everyone. So for the first part, the key message is that science is lagging very much behind reality. So cannabis is everywhere. There's a lot of products everywhere. They are diversifying. They're increasingly accessible. And yeah, a lot of people don't know out there how to quantify how strong is the cannabis that they have been using. So you can see an example of varieties of different cannabis products that we saw in Canada during a visit in early March last year. And part of the problem is that, I mean, there's a lot of angles to this issue, but part of the problem lies into poor metrics in research of cannabis and poor labeling of products to inform consumers, And also the fact it hasn't been very much a sexy topic. Indeed, this started with a bit of a story. When I was at the University of Liverpool, probably around 2018 already, I really wanted to pursue this topic on understanding and improving the measurement of cannabis use, because in our team, we did so many reviews on the risk and benefits of cannabis consumption. And one key thing that kept coming up in every single study or every single review is that consumption of cannabis use was reported very differently and a lot of times in ways that couldn't be integrated between the various studies. And that created a huge problem because we can't necessarily benchmark the risks of cannabis consumption. We can't tell if specific patterns of use or if specific dosages can actually exacerbate some of the health outcomes associated with cannabis consumption. And this is a bit of an example of some cannabis products that are out there, not in Australia, in Canada, in this case, but myself and another researcher, I'm going to write him out, Tom Freeman, we were at this meeting in Ottawa. And of course, we were very curious to see what does a legal market look like and what does a shop look like. So we went into a shop and asked to one of the people behind the counter, what would you recommend to somebody who doesn't use cannabis very much, an infrequent user or cannabis naive person? What would you recommend to them to use? And so this person came about and brought us this Mellow Man. So this is a little pack and it's got 3 joints, pre-rolled joints in there. And so, if you look at the cannabis dosage in there, the THC, which is the psychoactive compound, the main psychoactive compound of cannabis, it's about 4.5 milligrams per gram. But the thing is, when you do the maths and you see how big are the joints, people, if they smoked one of these joints, they would have 80 milligrams of THC, and you'd need about 5 to be intoxicated. So clearly this illustrates the point that there's products out there and people are not necessarily particularly infrequent users. They might not be aware of how intoxicating the product is, how intoxicated they're going to get. So effectively, people can't make informed decisions regarding what product they're consuming based on what the plans are during the day. So a notion that we've developed with Tom Freeman, and this was a side project, really, is developing the notion of the standard THC unit. And that's pretty much the equivalent of the standard drinks, but for cannabis. It's not supposed to be a perfect measure of intoxication. It's just supposed to be a metric of consumption, a quantity, a standardized measure of THC levels. And the reason why we chose 5 was because we looked at experimental evidence on THC administration in normative samples, and 5 milligrams THC produced robustly intoxication effects without minimal adverse reactions. Okay, so we decided to go with 5 milligrams in this instance. And so what was supposed to be a non-sexy project, completely unfundable, it turned into actually something influential, and that actually I haven't seen in my research prior, because I do more brain imaging research, where the implications for translations, of course, it's quite apart from it. It takes a lot of steps from biomedical research to have an influence. So it was really good to see that this got picked up by research funding agencies. NIDA endorsed the unit and recommended its use for research grant applications and in reporting research findings from research reports from the grants. And then it was also endorsed by neuroscience researchers and public health researchers and so on. So this really speaks to an existing, probably still unmet need for measuring cannabis dosage in a way that is standardized, that makes it easier for us to harmonize research findings across the globe so that we can join forces and produce better quality evidence. The other thing, and actually not just better quality evidence, but also it enables us to harmonize research findings between different studies, something that at the moment is lacking in the literature. Another initiative that took place was the CCSA in Canada, mandated by Health Canada, they were interested in exploring whether the THC unit is something that could be implemented in Canada, and that might be something different than 5 milligrams. It depends really on each country, what like the standard drinks. Exactly, the standard drinks are not identical in different countries. So in Canada at the moment, there are discussions in terms of how to better report the strength of cannabis products in products, and from a public health perspective, how do we inform consumers in a more transparent way. So at the moment, there are initiatives and reports in course, and hopefully Canada is going to lead the way for similar initiatives in other parts of the world. Another thing that, of course, came to mind was that the THC unit is something that Tom and I came up based on reviewing the literature on the topic. Nevertheless, it doesn't mean that 5 milligrams of THC is something that everybody necessarily has to agree on, being the best quantity of 5 milligrams. It shouldn't be necessarily the preferred quantity from all researchers or from different stakeholders around the topic of cannabis consumption. So what we have done at the moment, this is still findings in progress, we've conducted a Delphi expert consensus to ask the top researchers in the area of cannabis. We're talking about the top 60 Scopus researchers and other researchers that were recommended by them. And we are conducting currently a Delphi to see what researchers recommend should be reported to reflect the amount of THC in cannabis products. At the same time, we're starting a small survey for regulators, industry, and healthcare workers as well to ask for their opinion, and we are collecting data from consumers as well. And because we recognize, of course, that there's a lot of stakeholders in place and they all need to be considered. So the steering committee for the Delphi included Tom Freeman, Beatrice Carlini, Wayne Hall, and Dave Hammond. And some of the key items that we were administering were about how important do you think is to report THC content for the consumer on all products? And how important do you think it's to have internationally recommended metrics? And how how important do you think is to report THC content consistently across different methods of use? We're talking about bongs, vapes, and different methods, and edibles. How important do you think the units is consistent across all products and across different international jurisdictions? So definitely there was consensus across the first 5 items. This is actually just showing that the Delphi process, which is where we come up with items and then we get comments from all the experts, and then we have to ask the questions 3 times in a row and see whether the consensus is. And the consensus is established as 70% of people endorsing the item, or 80% if we can't, or 80% or 70% if we can't achieve 80%. So we reached consensus where everybody unanimously really endorsed that kind of THC content should be reported across in a similar way across different international jurisdictions, across different methods, across all products. Yes, for all the consumers and for products. Okay, so first of all, we established a consensus in that direction, which is actually quite important. Another set of questions that we asked consumer experts is also whether they felt like standard THC unit or percentage or total amount of THC would need to be reported in products. And we've achieved consensus of at least 70% for all of them in both rounds. And there's not really consensus regarding reporting other types of metrics in products or multiple metrics either. And also we haven't achieved consensus on reporting other cannabinoids, terpenes, or other contaminants as well. So the consensus here is around reporting metrics of THC unit, grams, and percentage concentration in products, and in a similar way across all products, jurisdictions, and modes of use. And this notion can be very helpful, hopefully, to guide consumers and policy practitioners to benchmark beneficial effects as much as risks as well. So just shifting to the next topic slightly. Another item is that we see all these beautiful graphs about the THC amount in cannabis products and how it's increasing in all world regions, except for in Australia. We don't really have data points or different points in times in Australia. So we see these beautiful trends and patterns, not necessarily beautiful, but in terms of from a research perspective, having multiple data across time is really, really informative to map whether the products are becoming increasingly intoxicating or strong, and what are the health implications. In Australia, we had a study conducted by Wendy Swift, which was published over 10 years ago, from cannabis collected in about 2010. About 10 years ago. I think they collected the cannabis about 2010. Okay, and similar to international trends, we've also seen that Australian THC 10 years ago had was highly concentrated in cannabis as well. And I'm about to show some results on some street cannabis results that we collected from Nimbin. So if it's possible to not take pictures of the results, because they're still ongoing, their early results. So what we've done, we, as part of a study funded by NHMRC, conducted at Monash University and led by Murat Yücel and Nadia Solowi, and they involved me in the process quite later. So I just wanted to acknowledge that I didn't create the study from scratch. They've done a fantastic work setting it up from an ethics perspective. It was very, very difficult. And what we obtained is really how strong it is, the cannabis that is consumed in the north of New South Wales. And this is data from a current cannabis plant matter. So you can see that the amount of THC is about 15% roughly, but this is the distribution. This is total THC. This is THCA, which is a precursor of THC, and this is delta 9 THC. Okay, and this is a concentration of other cannabinoids, but be careful that the axis here is up to 10, and this should actually be a very small portion. If you look at the axis here, they're quite different. This is up to 50, this is up to 10. So these portions, these levels are very, very low. And then we computed how strong was cannabis consumed by participants. We saw that participants consume about 1 to 2 THC unit per day. And we saw that the amount of THC was not correlated with people's perception of the strength of their own cannabis as well. So that's interesting how the perception of the strength might not necessarily match people's perception. Because we obtained this cannabis from regular consumers for a long time, it might be that the tolerance effect towards the effect of THC might somehow mask the individual's ability to just straight recognize how how strong it is, because tolerance process is in place. And Isabella Goodwin, our postdoc, has conducted the analysis. She's done a fantastic job. Wanted to acknowledge that. So if you compare our results, which are here, to the results from cannabis from about 10 years ago, and we're trying to keep similar axis. This is the axis 40. This is the number 40. You can see that, the amount of THC hasn't necessarily changed that much compared to 10 years ago based on these single data point. So watch this space. Of course, definitely there is a strong need to monitor and track Australian cannabis over time and across Australia. And that would be very informative, because we don't know what consumers are using at the moment in most of Australia as well, or whether cannabis is changing at all. Okay, so another issue that we've been investigating is also about, we are aware that there's no standardized metrics as well when reporting cannabis use patterns in research as well. So this is the kind of issue we see over and over in literature review. Okay, literature reviews. So as an example, one of our PhD candidates conducted a review a couple of years back, and she just looked at how frequency of use was reported, right, in the literature to date. So frequency of use was measured, some studies said no current use or yes current use, or number of participants with occasional daily use, max hit frequency in the past year, occasions per week, days per lifetime, days per week, days per month. Now, the thing is, we can compare, or we can cross-compare, we can convert days per week to days per month and the other way around, but nothing else can really be compared to each other. So that means we can't really integrate the findings from the literature to date to understand whether greater dosages are associated, or what type of dosage is associated with worse harm. Which is not to say there is, of course, merit to measure cannabis consumption in different ways, depending on the research question. But if we don't make an effort to standardize some key measures, that's going to be difficult to make advances in research. So as to illustrate an example, I can say I've eaten cake on 5 occasions this week. Wayne has eaten pavlova 5 days this week. You can't really compare whether we have eaten pavlova the same amount of times at all. So similar issues that not also apply to metrics of dosage, to metrics of age of onset. There's no need to go in detail in this slide, but this is just to illustrate this is a huge issue in standardizing cannabis consumption. And so what we've done a few years ago, we've created a bit of a consensus and invited about 70 people and cannabis researchers to discuss this issue, and with a number of presenters from different parts of the world. And the goal was to really identify the challenges in measuring cannabis consumption and what are the key metrics that every study, if we have to keep it bare minimum, if every study was to measure something, what would it look like, so that we can harmonize the evidence a bit better. So we came up with this framework with a universal layer implemented by all studies, a middle layer, which is context dependent, and biological measures at the very top of the pyramid. And the cost goes up as you go up in the pyramid, and the ease of implementation is easier at the bottom of the pyramid as well. And so in the bottom layer, there are just 3 questions, and I'm happy to go through this in detail later on. I'm just mindful of the time. But basically, we ask participants if they ever use cannabis, when they last used cannabis, and if in the past month, how many days they use cannabis. And again, it's a very rough measure, but at the same time, if all the studies ask for this question, we will be much further than where we are now in terms of integrating the evidence. The second layer is context dependent, and we propose the timeline follow-back methodology, which is a semi-structured interview that can be applied to 1 day, to more days, to 3 months, and you can go backwards as much as you can. And it's a validated, robust methodology that correlates with urinary metabolites of THC and cannabis dependence levels as well. A big issue when we measure cannabis dosage is really about how do we measure different modes of use, novel modes of use, such as gummy bears, vapes, wax, you name it, brownies, and so on, and so on. And so we recommend to use timeline follow-back methodologies to get as much information about the product type and the quantity utilized. And on the top, there are biological measures that go from very cheap and quick, deep tests to confirm the presence of cannabinoids, to blood samples on the most precise end, and THC levels in the product themselves as well. Of course, this depends on the resources of the project. So if some studies might just stop here, if we're talking about public health surveys, for imaging studies of the kind we conduct, because we basically kidnap people for a day, and participants are very happy to give information about the consumption. We have more time with participants, so we are able to get more data. And again, it depends on the type of study that the researchers conduct. Okay, so this methodology needs further validation and also needs to be complemented with more detailed population level monitoring as well. So hopefully this part of the presentation is helpful to reflect on perhaps some little tools that might be used in measuring cannabis consumption in research setting, so that our research in 10 years' time is a little bit more meaningful. It can be integrated with other findings in the literature. And a lot of people contributed to this work, so clearly this is not just my work. It would not be possible, so I just wanted to acknowledge that as well. As I say, I supported the work. The conference in Lisbon Addictions has supported part of the work, the MCDA and key funders as well. Now there's going to be a bit of a shift towards the brain imaging part. So if you have any questions on the metrics component, please, you might need to wait for another 15 to max 20 minutes. So when I came to Australia a long time ago, about 15 years ago, I was involved on some research studies, and this is a classic study that was conducted quite early in the days, where we looked at brain structure and the neurocircuitry of chronic cannabis use, and that was part of my early work together with Nadia Solowi and Murat Yücel as well. And something that we know is that according to neuroscientific theories of addiction, chronic substance use is associated with neuroadaptations in brain pathways relevant for stress, reward processing, and disinhibition. And in cannabis users, there hasn't been much evidence so far, and as part of this presentation covers some of the new findings in this regard. And one of the things that wasn't quite clear is whether, from a brain structural perspective, cannabis is associated with the changes in the brain through the brain, or whether it affects selected brain regions. So in this specific sample of just 20 people, 20 chronic cannabis users and 20 controls, we found that people who use cannabis and the whole sample show lower volumes of the hippocampus and amygdala, which are regions implicated in reward processing and a region implicated also in learning and memory as well. All the other regions of the brain that we examined, we didn't find any group differences. So a key message there from the get-go was that the impact of cannabis that cannabis might have on the brain could be regionally specific, and particularly in parts of the brain that are very high in cannabinoid receptors, which are the receptors in the brain that THC binds to in order to exert its psychoactive effects. So these regions might be more vulnerable to chronic THC exposure. And very interestingly, we can see that in the slide in the right, you can see the concentration of cannabinoid receptors in the brain. And on the left, you can see where the literature show the brain imaging, the volumetric reductions to be most consistently reported. So the areas that are a bit lighter, they are the ones where volumetric reductions are most commonly reported. Here, the darker areas show parts of the brain where we don't see many group differences. And interesting, there are some overlaps between where we see these brain differences to occur and the areas where cannabinoid receptors are located. Therefore, this lended to the hypothesis that perhaps chronic cannabis consumption through an effect of THC might have a select effect on parts of the brain high in cannabinoid receptors. Even if you look at acute intoxication studies as well, where brain function is looked at when people are intoxicated and not, we can also see that the same brain pathways are also implicated. So some alterations might affect structure and function as well. So we started building some evidence to try and understand whether these changes occur on everybody who uses cannabis or only on the people who experience problems with cannabis use. And this is quite important because according to these theories of addiction, the neuroadaptations would only occur in the transition from recreational use to dependence. And we also know that a lot of people who use cannabis, they don't necessarily experience any issues with their consumption, but a significant minority does. So these seem to be quite different groups of people. It's almost like thinking that somebody who uses alcohol recreationally would show the same brain circuitry as somebody who has chronic alcohol dependence. They're very different population samples. So that's where we started some pilot studies in the last few years, where we went back to our data and split the sample of people who use cannabis in those who did not report any problems with consumption and those who reported problems with consumption in various ways. Problems with consumption were measured differently in different studies. So we saw that for regions of the reward circuitry, we're talking about the striatum, nucleus accumbens, and the pallidum, we saw that participants who reported problems with use had different volumes compared to controls, as you can see, But people who didn't report problems with use, they didn't show any difference compared to controls. Same for the amygdala volumes as well. People with problems with higher problems with use, they show lower volumes than people with lower problems with use. And we found similar results when we looked at other brain regions that are implicated in addiction, such as the cerebellum, such as the hippocampus as well. Again, you can see that the differences are only between dependent use consumers and controls. And this is work conducted by Dr. Yan Chi. And when we combined data between different parts of the world and different data sets to get greater sample sizes, we confirmed exactly the same, that the only difference was between controls and people who report dependence on cannabis. There's no difference between controls and people who don't report dependence on cannabis as well, okay, and who still use cannabis. And some of the recent work that we've done is also to try and see and focus on people who report use problems with cannabis. Then, based on these findings, we're like, okay, let's focus on understanding better using the DSM-5, what's happening with people who report problems with cannabis. So we looked at cue-induced craving in people with moderate to severe cannabis use disorder in a number of different samples. And cue-induced craving is important because it's associated with it's associated with relapse, with chronic use, and knowing that there's a lot of cannabis stimuli in the environment at the moment, it's very important to understand how looking at cannabis stimuli affects the brain circuitry implicated in craving. So we've conducted a series of 2 studies. One was conducted in Wollongong, and we recruited people who are young, mostly university students, with a range of CUD severity compared to controls, who used at least 4 days a week and who used between a gram or 2, about a gram a day, up to a gram a day, as you can see here. And people were intoxicated at the time of scan. And we found greater activity in brain pathways implicated in sensory processing, reward processing, and salience in young people with a cannabis use disorder compared to controls. Then we did some research in Melbourne as well, and we focus on people in the community, in adults who try to cut down or quit, but they're not looking for treatment, and they have moderate to severe cannabis use disorder. So this sample is a bit more adult, but they still were using every day or almost every day. And what we found, again, we found a more marked pattern of greater activity in brain pathways implicated in motivation, salience, reward processing, and disinhibition. And this is work conducted by Dr. Hannah Sales from our lab. And also she collected data during COVID, so that's why the picture's there. It was really difficult to run. And we saw that this brain activity correlated with greater withdrawal, with greater arousal. When people looked at cannabis pictures, people experienced greater arousal and with greater levels of THC metabolites in urine. So we thought it was very interesting, and we thought, okay, so maybe, is there anything we can do to reduce the reactivity of the brain during cue-induced craving? So we started implementing we started trialing different strategies, and so I'm going to report some preliminary findings. So we did a mindfulness-based intervention, a double-blind, active placebo control. We conducted an RCT. It was really difficult again during COVID, but we really looked at the brain regions that we know show greater reactivity in people who use cannabis. And we found that the mindfulness-based group showed reduced activity in the middle temporal gyrus, which is a part of the brain which is implicated in stress and learning and memory as well. And the other active placebo intervention and passive placebo intervention, we saw no change pre to post these interventions. This is a brief intervention of about 2 weeks that people had access to online for 2 weeks, and we monitored their practice, and then they came a baseline and a follow-up. So perhaps there's something into these mindfulness brain strategies. It's not just a generic intervention, it's an intervention that was shown to reduce craving and that targets specifically craving for a few minutes a day, and was demonstrated to reduce craving in alcohol dependence, in risky users, actually, risky drinkers. We're also designing some new neurofeedback studies where people can receive brain feedback in real time on the brain activity when they're craving. And these includes PhD students such as Amir and Ethan from the lab and a research team, because from a technical perspective, it's quite important to design the experiment correctly. So we hopefully in future I'll be able to present some of these findings as well. And we have some preliminary findings from a cannabidiol trial, where cannabidiol was given to people who use cannabis for a few weeks. I think that it was for 12 weeks, for 10 weeks. And people were given 200 milligrams of CBD every day for 12 weeks. And what we found here as well is that in people who use cannabis, there were significant changes in the function of key regions. And so in the hippocampus and presentral cortex, the hippocampus is implicated in stress, learning, and memory. And in the amygdala, connection with the lingual gyrus. The amygdala is implicated in craving, and the lingual gyrus is implicated in the salience processing of visual stimuli. So these are, again, preliminary findings that, of course, require replication in larger RCTs. And last but not least, we have some findings from a study called the Imagine, which is a huge consortium on youth. There is about 2,000 youth scanned at age 14 and at age 19, and then it kept going from Europe, multiple sites. And what we had is we isolated a small sample that used cannabis at age 19, but not in a sample of controls that didn't. And we matched these two samples by anything you can imagine to make sure the groups were well matched. Then what we did, we went back and we looked at the same sample before the cannabis use commenced to understand whether any of the differences we might have seen in brain function predated cannabis use onset. And again, all the samples were matched for all sort of variables, including substance use. However, the young people who use cannabis, who ended up using cannabis, had a greater level of impulsivity, which makes sense. So what we found is that there were group differences between cannabis users at age 19 and youth who didn't use controls in a variety of brain pathways. A lot of these pathways here at the end are visual pathways implicated in salience processing again, and we can see ventral prefrontal cortex regions that are really implicated in motivation and again disinhibition. And we found that these group differences at age 19 correlated with impulsivity levels, and none with cannabis use levels. So these findings suggest that perhaps in infrequent cannabis consumers who are young, age 19, these differences might be driven by impulsivity, not necessarily by the use of cannabis. And the postdoc driving this project is Sampras Ganesan, who's recently finished his PhD, and it's fantastic. He's going to have a great career ahead. So what we found is that at baseline, at age 19, we didn't really find any group differences. So look, we can't infer causality ultimately, but we just know that these brain differences weren't there in the groups before the onset of cannabis use. Right, and these last two slides are about more future directions and some of the future directions. So we know that there is a bias in the research, in substance use research, towards an overrepresentation of male samples, which is shown here. So if you look, for example, just at imaging studies, imaging of the brain, any data point that is on this line includes samples that have an equal number of males and females. All the data points that are down here, they include more males than females, and the samples here include more females than males. So overall, it's very apparent that the evidence overrepresents males and what happens to females, but also when we try and understand the core mechanisms of substance use, are these mechanisms related more important for males or females or for both? We don't really know at this stage, because also a lot of the studies to date don't account for sex differences appropriately from a statistical perspective either. For example, sex will be used as a covariate rather than as a predictor or an interaction term, together with the substance abuse. So we conducted a Delphi consensus with researchers at NIDA and people from about 37 different world regions as well. We're just coming up with a bit of a consensus statement to move the field forward. And if you're interested in collaborating on this topic or joining forces, please let us know, because it will be fantastic to have more people on board. Another new study that we're actually recruiting new sites for is we created a new Enigma consortium, which is called Enigma Cannabinoids, together with Dr. Lisa Greenwood at the University of Wollongong. And in this mega study, we are collating data sets from brain imaging studies where cannabinoids are administered medically or experimentally. So we're hoping to then unpack and join forces the fundamental mechanisms of cannabinoids action, because at the moment the evidence is quite mixed. So we need bigger numbers. So that's pretty much it. These are the key take-home messages as well. Probably account for gender and sex differences if possible. We need new treatments for cannabis use disorder, and we need to measure cannabis use perhaps a little bit more better in a harmonized way to create robust evidence. So thanks so much, and thanks everyone who contributed to this work in this slide. And thanks to the team in my lab. They work really hard every day. So yeah, thanks everyone.
Fantastic. Thank you so much, Valentina. That was such a broad, all-encompassing scope that was then funneled down into the nuance involved in cannabis use. And obviously it's built on such a long history of research, but it's come at such a good time as we can see that there's a growing acceptance of cannabis, both publicly and legislatively. I can see that we've received a number of questions already, so I just want to give a reminder, please pop the questions into the Q&A function. But before we answer any questions, I would like to throw over to Professor Wayne Hall to provide his reflections on that amazing presentation.
Thanks, Calvin. I'll largely concentrate on the first part of Valentina's talk, which is around improving quantification of cannabis dose. And there's obvious reasons for this. Dose matters for all drugs. Generally, harm increases with increasing dose, although not necessarily linearly, but it typically tends to be monotonic. That is, as dose increases, harm increases, or it might level off. And to date, certainly most of the time that I've worked on cannabis use epidemiology and harm, we've had limited measures of quantity. We've typically tended to use daily use as a proxy for high doses of cannabis. And that's probably been a reasonably defensible, as Valentina has shown in the second half of the talk. It's the daily users who are much more likely to be problem users who tend to show more of the problems associated with the use. They tend to use more often, they develop tolerance to the effects of THC, and they tend to use cannabis more frequently, that's per day, than do less frequent users. And so they typically have tended to attain much larger doses of THC than people who might use, say, less than weekly. So it's been a reasonable proxy. And while the characteristics of cannabis products was gradually changing over time, as we know that THC content of illicit cannabis in most jurisdictions, the US and Europe, has gradually increased over time. What's worth remarking, I think, is that even the crude measures that we have had to rely upon for dose have been sufficient to detect adverse effects of cannabis use. And the same is true. We should, I guess, remember that the epidemiology of tobacco smoking has used similarly crude measures of cigarettes per day, despite the fact that people smoke tobacco in different forms and with different contents of nicotine and tar and other nasties. The same is true for alcohol epidemiology, with the drinks per occasion and days per week of use. These sorts of noisy measures are sufficient to detect associations between particularly daily and heavy use of these drugs and adverse effects. But the noise in the measures probably are more likely to underestimate the strength of associations rather than vice versa. But as Valentina has reminded us, dose matters greatly when the cannabis market is changing, as it is rapidly now. There has been a steady increase in the THC content of cannabis products on the illicit market. And what we've seen, I guess, a bit of a surprise to many of us is how rapid the proliferation of high THC content cannabis products has been, initially in the medical cannabis markets and now in adult or recreational cannabis markets in Canada and the US. And so we see cannabis flower with 15–20% THC content sold. We have fortified joints being sold in Canada where THC is added to herbal cannabis to produce joints with 30% THC. And we have the extracts that concentrate THC to levels 70% and higher. So that's been a big change. And John Corkins, the drug policy analyst in the US who's worked at RAND for many years, has done some simple calculations to indicate the huge change in typical dose of THC that people have consumed. He took the example of someone who had a single joint on a weekend back before 2000 in the US, when a typical joint was sort of about 4% THC, and they were getting around about 5% milligrams of THC from that particular joint. And he's estimated that an average daily user now, and these are increasing in prevalence in the US since legalization, it might be consuming 20% THC products containing 20% THC and smoking several joints per day and getting up to 320 milligrams a day of THC. So a huge difference in THC dose over a relatively short period, accelerated, happening in the illicit market, but greatly accelerated with the legalization of cannabis. Now, the challenge is, one of the arguments, of course, for legalization was that you could regulate cannabis, ensure that there was standardized dosing, and hence ensure that consumers were better informed about what they were using and how much THC they were obtaining. And clearly, this states and jurisdictions that have legalized cannabis have made some form of labeling obligatory in the interest of consumer protection, as they should. But as Valentina indicated in one of the studies she's doing, the Delphi, which I'm involved in, it's not clear how best to label the THC content of cannabis products. Even when we're looking at a single product like a joint or an extract, should it be the percentage THC, the milligrams of THC per dose, or the total milligrams? A variety of ones. There are empirical questions that should be a priority in terms of identifying the best way to provide consumers with information about the products they're consuming. There are major challenges, of course, in specifying a standard dose. The pharmacodynamics and pharmacokinetics of oral versus inhaled routes of administration widely differ, as do the typical doses that an infrequent or very frequent user might consume. So it's not clear what the ideal dose might be there. And I think the 5 milligram dose that has tended to be arrived at is probably a sensible one that's erred on the side of precaution in specifying a dose that would be sufficient for the majority of infrequent users to obtain a desired level of intoxication without increasing the risk of adverse side effects. But the challenges in specifying a standardized dose are not unique to cannabis. We've had the same sorts of arguments in the alcohol epidemiology area about what a standard drink is. The grams of ethanol between what's called a standard drink in even different English-speaking countries differs fairly widely. And as I've mentioned, the sort of tobacco products, this is before e-cigarettes and nicotine patches. We're reproducing in the nicotine area a lot of the same uncertainties about specifying dose we're now seeing or wrestling with in the case of cannabis. It's worth making the point that the field of pharmacoepidemiology, the study of the use of drugs in the population, typically prescription drugs and medical or pharmaceutical drugs, has tended to use a defined daily dose, which is analogous to the sort of standard dose that we're talking about with cannabis. And it's recognised that that has its limitations as a way of measuring, but it's a way of indicating, typically for a drug, the standard dose required to achieve a therapeutic effect in an average sample of people. And that's what we're looking for in the standard dose for cannabis. I think I'd end by sort of making the point that whatever the standard dose is or the measures we use don't have to be ideal to be useful, and we shouldn't let the ideal be the enemy of the good enough when it comes to specifying standard doses. Their value will vary clearly with their purpose, whether it's to inform consumers about what they're using, for laboratory and clinical studies of the effects of cannabinoids, or epidemiological studies of harms related to cannabis use. These are all empirical questions, and our decisions about the best way of specifying these things are obviously open to revision in the light of research, and we clearly need to give priority to this, given the increasing move, the sort of bandwagon towards legalisation of adult use that we now see in many high-income countries and in some low-middle-income countries such as Thailand, Mexico, and South Africa. I'll leave it there so that there is some time for questions from the audience and comments. Thank you.
Fantastic. Thank you so much for that reflection, Wayne. You actually touched upon quite a number of questions as well at the same time. Maybe just for a little bit of clarification, either Valentina or yourself, Wayne. Margaret White wants to know if the 5 milligram, and I know you had mentioned this, in each route is relevant to each route, so oral versus smoked, for example.
Yeah, the effects will be comparable, but there is a different timing whereby the onset of edibles is later. But one of the issues is that kids or hospital personnel, hospitality personnel might think that they're normal lollies and ingest it and then forget about it and then get intoxication when that's quite scary and end up in emergency and all of that. So I think that yeah, but it's the same level of intoxication, just different set. Wayne, anything else from your
Well,
end?
I mean, I think just the only point you'd make is to make clear to people the very different time onset and duration of effect from oral use. And clearly, if we look at the major adverse consequences of legalisation, the biggest one has been an increase in emergency department attendances for overdose of particularly oral products, both amongst children, but also adolescents and young adults.
Absolutely. Makitsa, sorry if I'm pronouncing that wrong, has highlighted that the Australian HREC guidelines for medicinal cannabis are far higher than the 5 milligrams that you suggest, up to 500 milligrams daily via inhalation and 40 milligrams orally. They're just asking around whether this single dose unit does it equate to the use of an alcohol unit in the sense that it's quite low in comparison to guidelines?
Well, 5 milligrams of THC is just based on experimental evidence of intoxication. So based on evidence, it's not a number that we just came up randomly. It was about what does the evidence say produces robust intoxication in infrequent users. I think it also speaks to the point of communicating to consumers in a clear way of medicinal or non-medicinal cannabis equally, what level of intoxication they might expect from the products they consume. I'm not quite sure about if it's perfectly equivalent to the level of intoxication from alcohol, but it's definitely informed by evidence of intoxication in normative samples. I don't know, Wayne, if you have any other notes or comments.
Well, the only thing I guess I'd say with the medicinal is the sort of doses that are now being prescribed, I think is sort of an evidence-free zone. I guess for some of the indications for chronic pain and so on, people will develop tolerance and so require higher doses. And so the analogy here, I guess, would be between a defined daily dose of a drug like morphine, which might be used to relieve pain in an opioid-naive individual, would be a tiny fraction of the dose of morphine that might be administered to someone who's been been treated with morphine or another opiate on a regular basis, say for chronic pain or or a pain of malignant origin, say in the case of cancer. So you wouldn't expect these sorts of doses that are primarily, I guess, designed for informing recreational non-medical users to apply straightforwardly to medicinal ones. But I think some of the THC doses and the total quantity of cannabis that's now being dispensed in the medical cannabis program in Australia would be really hard to justify from, pointing to evidence from clinical trials of efficacy. Because I think a lot of the clinical trials would not use doses of the sort that are now being prescribed. And I think the TGA is concerned about that. And so I would expect there to be some sort of pushback from the regulatory authorities about what's happening in the medical cannabis market.
Yes, and I think obviously it sounds like a simple question, how much is one standard unit? But the more you dig down deep into it, the more questions that arise and it becomes very complex. So to even just have a value like 5 milligrams is just a good starting point to start those conversations and discussions around consensus. Mark Powell is interested in first episode psychosis and looking at the comparison of youth cannabis use in conjunction with first episode psychosis, and if you know anything about brain changes in relation to that.
The evidence is still a little bit complicated or unclear because usually you need a sample from a design perspective with psychosis controls, and then people who use cannabis alone and people who use cannabis and also have psychosis. So still the evidence is relatively early. Nevertheless, it seems like there's some marked changes in early episode psychosis in cannabis users. However, it's also not clear how much some of the changes are due to medication that people might take in first episode of psychosis as well. That could change also the brain. So I'm afraid I don't have a specific answer based on my understanding of the evidence. There seems to be some interactive effects. Nevertheless, it's still unclear what the role of the medication is. I don't know, Wayne, if there's anything from your perspective.
Well, I guess in the epidemiological perspective, Benedict Fisher and colleagues recently published a paper reviewing the epidemiological evidence, looking at what's the sort of crucial point that increases the risk of a psychosis amongst cannabis users, and that it turns out to be daily use, particularly daily use. If young people initiate early, that is in mid-teens typically, they get involved in daily use and they're using daily over substantial periods of time, they seem to be a particular high risk of developing chronic psychotic illnesses. And I think the point to be made is that a lot of young people who have this pattern of use engage in it for some years before they end up presenting with symptoms of psychosis. And the real worry at the moment, I know it's largely anecdotal, has been the number of psychiatrists reporting patients with psychoses who are getting prescriptions of high THC cannabis, medicinal cannabis, which is meant to be contraindicated. I think the evidence is fairly clear that people with psychotic illnesses who engage in regular cannabis use do a lot worse than their peers who don't. And it's certainly something that we should be strongly discouraging.
Thank you so much for those answering all those questions. Unfortunately, we weren't able to get to as many as I'd hoped, and I'm sure you would love to answer them all. If we didn't answer your question, please reach out to the NDARC communications team, or if you have desire for any of the resources. I've seen questions around what measures were suggested, for example. We can put you in touch with Valentina or answer on their behalf to facilitate that communication. Unfortunately, we're going to have to say goodbye for now, even though there are many unanswered questions. But thank you so much, Valentina and Wayne, for joining us today and for presenting such an amazing breadth of work, and for providing your reflections, Wayne. Looking towards the future very quickly, on the 5th of March, we will have NDARC's Bianca Varni presenting on the use and safety of prescription opioid analgesics during pregnancy. So there's a bit of research focused on with a woman perspective as well, Valentina, as you mentioned. So hopefully we see more research that highlights populations that include women, if not equal or more. And following that, on the 12th of March, we'll be looking at Jack Freestone, another person from NDARC at NCRED, exploring and implementing peer-led chemsex interventions for gay and bisexual men. So another population of interest, two very interesting presentations that we hope you can join us for. So thank you again for joining today, and thanks, Valentina.
Thanks, everyone. Thanks, Calvert. Thanks so much, Wayne. Have a good day.
Thanks.